Mhm. Hello, everyone, and welcome back to the Swalife Podcast Series. If you think about a drug discovery, almost every big idea begins its life in a lab dish. Before animal studies, before clinical trials, and before headlines, there's always an in vitro experiment that decides whether an idea deserves to move forward at all. And yet, even today, many in vitro studies are designed based on habit, past experience, or this is how it's always been done. That is starting to change now. Artificial intelligence is not just helping analyze in vitro data anymore, it's beginning to shape how these experiments are designed in the first place. In today's episode, we are going to unpack what in vitro studies actually are, what it really means to design them using artificial intelligence, how this changes failure detection, and how AI studios are building structured approaches around this shift. I am joined by Dr. Pravin Badhe. Welcome back, sir.
Thank you, Shruti. This is an important discussion because when we look at in vitro studies, quietly help us to determine the success or failure of the most discovery program, often longer before anyone realize it.
Yes. So let's begin with what in vitro studies actually are, and why are they considered so foundational in biomedical and drug discovery research?
Yeah, because in vitro studies are experiments conducted outside a living organism. So typically in controlled laboratory environments such as cell culture or biochemical assays or enzyme system or even in the engineered tissue models. So their foundational role actually comes from the fact that they represent the first controlled interaction between a scientific hypothesis and the biology. So at this stage, we are not trying to demonstrate clinical efficacy. So instead of we are actually asking much more basic, like crucial questions, like does this molecule interact with the biological system we think it should? Does it influence the pathway or target we are interested in? Does it show sign of toxicity or non-specific effect even before we get to complexity? So here, in vitro studies act as a biological filter. They help to eliminate ideas that are chemically elegant but biological irrelevant. So they also help to refine promising ideas before more expensive or ethically complex studies are undertaken. So without a strong in vitro foundation, downstream studies often fail, not because the science was wrong, but because the early biological assumptions were never properly tested.
So would it be fair to say that in vitro studies are more about direction setting rather than proving success?
Absolutely. In vitro studies rarely prove that the drug will work in human. What they do is set direction. They tell us which hypotheses are worth pursuing further. They also inform us that which biological mechanism seems actionable. And also, the important part is that which idea should be stopped early. In that sense, in vitro studies are less about validating and more about navigation. So they help to guide where resources, time, and also the effort should be invested next.
That's a great way to put it, and it's really about choosing the right path early on. So, when we say AI designed in vitro studies, what does that actually mean, and how is this different from the way experiments are usually planned in labs?
So if you look traditionally, in vitro experiments are designed by looking backward. So scientists rely on existing protocols, published literature, and personal experience to decide what to test. But when we say AI design into in vitro studies, basically we are changing this by looking forward. So instead of starting with the protocol, AI will start with uncertainty and decision points. So here it's going to ask, what do we still not known about this biology? What experiment would reduce uncertainty the most? And it will also ask what outcome would actually change the next decision. So AI system analyze the large volume of historical experiment data from the literature, also the biological pathways, and also the outcome patterns, including the failures. That's the important part. So based on that, it is going to recommend experiments that are decision relevant and not just technical standards. So this does not mean that AI runs experiment or replace the scientist. It means that AI help decide which experiments are worth to run at all.
So AI is not telling scientists how to do experiments, it's helping them decide what's worth testing.
Exactly. So here AI does not replace the lab skill or biological intuition. Instead, it acts as a prioritizing layer. So in many programs, the biggest inefficient inefficiency is not poor execution. It's a running experiment that don't meaningfully produce informed decisions. So AI help to focus efforts on experiment that actually move forward.
That really shifts the conversation from how to why we run experiments. So, at what stage of drug discovery do AI designed in vitro studies add the most value? And why is timing so critical here?
AI designed in vitro study add the most value early when assumptions are still feasible uh, and also decisions are reversible. Early discovery is full of unspoken beliefs like what is the target or is the target relevant, that a pathway is actionable or the modern system is representative. So all these assumptions and beliefs are there. AI helps surface and test these assumptions quickly. So by designing in vitro study that directly interrogate biological relevance, AI helps team avoid building entirely program on weak foundation. So the earlier this uncertainty are addressed, the less likely a program to fail later for fundamental reasons.
Is that why so many late stage failures can be traced back to early in vitro decisions?
Yes, very often when we look the late stage failures are rarely sudden. They are usually delayed consequences of the early assumption that were never challenged. So if early in vitro study don't fully test relevance or robustness, those gaps tend to resurface later when correction is much more costly.
That really highlights how much responsibility those early experiments carry. Now, in practical lab terms, how does AI help optimize things like dose ranges, time points, and biological readouts in vitro assays?
So traditional in vitro experiment often try to cover everything. So when we look at the in vitro studies, many study covers things like many doses, many time points, many readouts, and that is because of the uncertainty is high. AI approach this differently by learning from the prior data to predict. So it will help us where those response transitions are likely to occur, which time points are more informative for a given mechanism, which readout are most likely to correlate with the downstream outcome. So instead of maximizing coverage, AI maximizes information per experiment. So this leads to experiments that are more focused, easier to interpret, and more closely tied to the decision.
So it's not about doing pure experiments, but doing more meaningful ones.
Exactly. So AI does not aim to reduce uh, ambition; it reduces the noise. So by focusing on experiments on informative reasons, teams can learn more with the fewer iterations, and it will avoid down drawing into uninterpretable data.
So can AI designed in vitro studies help detect failures earlier, and how does that change decision making for the R&D teams?
Yes, and this is one of the AI's most valuable contribution. AI systems are trained not just on successes, but on pattern of failure. Things like inconsistent responses, early toxicity signals, or lack of robustness across conditions. So this allows AI to recommend experiment that intentionally trace a hypothesis. So instead of asking, does this work under ideal condition, AI is going to help to ask, does this still hold when the condition varies? So early failures allow team to stop weak ideas sooner and redirect resources and avoid failure confidently.
So in a way, is it true that AI helps teams fail faster but also fail smarter?
Yes, exactly. Failing early with clarity is far better than failing late with regret. So AI is actually going to help to transform failure into structural learning rather than wasted efforts.
So yes, failing smarter definitely sounds like a progress. With AI helping design experiments, where do human scientists still fit in?
Okay, so with human scientists, they are still central to the discovery because AI can suggest experiment, but humans are the one who decide which biological questions are meaningful. They are going to interpret ambiguous or conflicting results, and they're going to apply the contextual knowledge that data alone cannot capture. So biology is rarely clean or binary. So judgment, experiment, and intuition remain essential, especially when data is incomplete or contradictory. So AI support reasoning, but responsibility and accountability still stay with humans.
That balance between AI support and human judgment feels really important. So let's talk about what Swalife does. Swalife has developed two AI studios focused on discovery and study design. So how do these studios approach in vitro studies, and what makes their workflow different?
Okay, with our approach, we actually build around structured scientific prompting, and we are not doing automation. So the AI Discovery Studio work upstream. So it focus on understanding disease biology, pathway relevance, and the target context. So this ensure that the in vitro studies are grounded into meaningful biology before experiments are planned. So the AI Discovery and Study Design Studio formalized in vitro design. It generates structured prompts that define biological question, the experimental objective, the minimum assay set requirement, and how results should connect to the next discovery step. So together, this studio ensure in vitro studies are not isolated experiment, but part of the continuous experimental scientific narrative.
So instead of asking what assay should I run, scientists are really being asked what decision am I trying to make?
Sorry, can you repeat the question?
Yes, sure. So instead of asking what assay should I run, scientists are really being asked what decision am I trying to make? Am I audible, sir?
Yeah, yeah. So can you repeat it again?
Yes, sure. So instead of asking what assay should I run, Scientists are being asked, what decision am I trying to make?
Yeah, so okay. So exactly that a shift from essay selection to decision clarity is the core innovation. So when we look at this, it reduces the redundancy, improve the reproducibility, and also keep experimental experiments which are aligned with the long-term goal. So that's the important part in the core innovation.
Yes. So looking ahead, how do you see AI designed in vitro studies changing day-to-day wet lab work?
Okay, so when we look at the wet lab will become more decision focused. So instead of just running the large numbers of loosely connected experiments, labs will run tight experimental sequence where each step inform the next. So AI is going to help to reduce the redundancy, the improved reproducibility, and shorter feedback loop between data and the decision.
So this means smarter labs and not just busier ones. So finally, let's talk about careers. What kinds of opportunities are emerging in AI-driven in vitro research, and what roles will define this space?
Okay, so we are seeing a shift from task-based role to decision-centric roles. So if I mention some of the emerging profiles here, so like experimental strategic scientists, so focusing on designing experiment sequence that resolve the biological uncertainty efficiency, often work across AI and wet lab teams. So that's where the experimental strategy scientists come. The next can be AI biological translator. So these are the one who interpret AI outcomes in terms of the biological. And they also ensure that the recommendation are experimentally meaningful and the context aware. That is another. Then the third one, it can be early risk intelligence scientist who can actually design experiments specially to surface biological, toxicological, or translational risk before moving into early drug discovery. Then the fourth can be the discovery continuity lead, where maintains coherence across discovery stages, so that way they can ensure that the early in vitro assumptions remain valid as program evolves. So these are some of the roles which sit between the disciplines and are increasingly critical as we see the increasing AI execution.
This really reframes career growth and stewardship of decisions and not just data generation. So today's conversation really brings home one simple idea. In vitro studies have always been the backbone of discovery, but how we design them is finally evolving. AI is not here to replace wet lab science. It's helping us ask better questions earlier, choose smarter experiments, and learn faster from both success and failure. What really stood out for me today is that the shift is not about running more experiments or moving faster at any cost. It's about being more intentional, about making sure every experiment earns its place in the pipeline. Thank you, sir, for walking us through how AI designed in vitro studies are changing the way scientists think, plan, and decide.
Yeah, thank you, Shruti. When we see the real opportunity here is not automation, but clarity. Clarity is how we connect biology, experiment, and the decisions.
Yes. And to everyone who is listening, as AI becomes more deeply embedded in discovery, the future won't belong to those who run the most experiments, but to those who design the right ones at the right time and for the right reasons. That's all for today's episode of the Swalife Podcast Series. Thank you for listening, and we will see you in the next episode. Thank you.
Thank you.